All right, welcome everybody. The Office of Programs to Enhance Neuroscience Workforce Diversity within the National Institutes of Neurological Disorders and Stroke is very pleased to welcome you to the Open Stage webinar series. This is a targeted outreach effort that we have for our diverse neuroscience community to learn about NINDS programs and network and connect with NIH and NINDS program staff. So this month, we are very excited to welcome the NINDS staff and grantees from the Office of Neural Exposome and Toxicology, or ONETOX. So before we kind of get started officially, I just have a few meeting reminders. You all are muted. This is a webinar, so you won't be able to raise your hand or anything. But please submit your questions using the Q&A function at the bottom. You can upvote questions and we will ask them at the end. You can enable closed captioning under live transcript at the bottom of your screen. And then also as a reminder, this meeting is being recorded and it will be transcribed and archived on our NINDS website and YouTube page. So we will share details about where to find that archive version of the meeting. We'll share the slides and we'll share all the resources that we talked about today with you in an email at the end of the week. So you will get everything at the end, don't worry. We do hope that you stay connected with us. Feel free to email us if you have any questions about this webinar series. And with that, I'll pass it off to Dr. Chardell Spriggs to get us started.
Thank you, Dr. Ebrahimi. Good afternoon, I'm Dr. Chardell Spriggs, and I'm here to welcome you to our webinar about exposomic research at NINDS. We've put together a dynamic panel that will shed light on this rich and complex area of research. Panelists will give a high-level view on the type of research we're interested in at NINDS, introduce some funding opportunities, as well as anchor the neural exposome concept and how it may relate to you. And hopefully, after this webinar, you all will be inspired to take a leap into neural exposome research. First, we will hear from Dr. David A. Jett, Director of the Office of the Neural Exposome and Toxicology here at NINDS. He will set the stage with an overview of the office, define the neural exposome, and orient you to our relevant mission space and opportunities. Then we will have Dr. Erica Glasper, Associate Professor at The Ohio State University in the Department of Neuroscience, share her research, journey, and insight with you. Following Dr. Glasper's section, we will have what I know will be a robust discussion moderated by Dr. Neil Drew. Some of you have submitted your questions beforehand, and we'll get to those during the Q&A, but I encourage you all to not hold back. Please put your questions in the Q&A box as you think of them. And with that, I'll turn it over to Dr. Jett.
Hello, thank you, Chardell, and hello, everyone. So we have a lot of people registered for this webinar. It's on the neural exposome and what we are doing at the National Institute of Neurological Disorders and Stroke at NIH. And really the importance of this in terms of health disparities and inequities. So just as a, to let you know about NIH, if you don't know, it has many different institutes. It's based on their focus. Our focus is on the brain and the nervous system. And our job is to reduce the burden of neurological diseases for all people. So I'm Dave Jett. I'm director of this office of Neural Exposome and Toxicology. Been here for more than 20 years. Before that, I was faculty at Johns Hopkins as a neurotoxicologist. I just wanted to also just give a shout out to all of my HBCU comrades and especially my fellow Hampton University Pirates. I had to put that in. So let's continue. Let's see. All right. So our office, it's the Office of Neural Exposome and Toxicology. It's called ONETOX for short. And it has 3 branches, but we're only going to talk about the neural exposome. And I have a fantastic small group that runs the office, but you'll see it's not surprising that we work with all of the staff at NINDS and many other institutes and centers with different foci at NIH. So thank you to the scientists, Dr. Sprigg, Dr. Drew, Dr. Figueroa Romero, and our fabulous operation coordinator, Scarlett Sella. And I'll just say again, 3 of the people you see here are from HBCUs. So we're real happy to have a lot of HBCUs working with us here at NINDS. All right, so the exposome. So I just want to say that as I go through these slides, I'd like you to think of why exposomic factors are likely the primary cause of health inequities. So just think about that. So exposome, so we know that genetics has, in the genome work, has provided valuable insight into the etiologies of these inherited diseases. But the fact of the matter is most of these risk factors can't be explained by genetics alone. And so a while back, Dr. Wild in a paper coined this word exposome. This is back in 2005. And really at that, he was talking about environmental exposures affecting the genome. Now it's actually all non-inheritable factors, not just environmental exposures, that affect gene expression, and not just across the lifespan. So now this is, I think, a new frontier of biomedical research to complement the really remarkable work in the genome. So what do we want to do with this? Well, we want to unlock a more holistic approach to disease prevention and more effective and personalized interventions. So that's just a general thought of the exposome. Let's see. So this is a paper that we did in Neuron. We know that, many, not most, neurological disorders have a complex etiologies that do include some of these non-inheritable factors, and we call it the neural exposome because it's focused on the brain. So what are these factors? So we have it, and everybody, there's a lot of different pictures like this that go around when they describe the exposome. And we think that there's 3 major things. There's exogenous factors like environmental toxicants, exposures to environmental toxicants, climate change, noise, endogenous factors, of course, like epigenetics, also metabolism, microbiome is big with us right now. We've been working on that. And then we call it behavioral factors, which does include psychosocial effects, but also includes things like lifestyle, sleep, very important, and very important, stress. So those are what we are looking at when we're talking about the neural exposome at our office. So, we do know that there are neurological health inequities that need to be studied, and more importantly, they need to be fixed and not just studied. You can see on this, you can see the Alzheimer's disease examples on this slide. So Alzheimer's disease is twice as likely in Black people than white people, and this is just not acceptable. I believe at least that the rise of more exposomic research will help figure out which exposomic factors are most important and which need to be addressed. And I think that's going to move a long way for these health inequities. Okay, so a lot of talk, but what are we doing? What is our office doing? I would say that we've made a good start. The office is only 2 to 3 years old, and we have already had several funding opportunities and grants that are funded. The ones with the red stars on this are the ones that are still active, and please, we would be happy to talk to discuss with you about these. So the examples are, we did work on environmental toxicants in Alzheimer's disease. We work with the ECHO program, environmental influences on child health outcomes. We have some work on climate change and health, which is a big, I think a big issue right now, especially what we're seeing in the climate. We've worked for a long time, actually, since day one, on biopsychosocial factors, specifically on social connectedness and isolation. We have one that's out right now. It's a pretty complex one, but I think important one. It's on the role of environmental stress in the health inequities we see in Alzheimer's disease-related dementias. We had a great one on the microbiome, got a lot of people interested, and we're funding some really interesting programs on that. And pain research is big at NINDS, and we've had something with them. How does this exposome affect pain? And then we've had some administrative supplements, and we're also working with EHS on what is going to be a new center for exposomic research to sort of coordinate what we're doing. And I think that's going to be interesting. All right, I'm almost done. So we've had a lot of outreach to try to engage more people to help bring, in our case, more neuroscientists together with toxicologists, because exposome just will not work if we do not have experts working together. For example, we would need an expert in Parkinson's disease working with a toxicologist that works on pesticides. That's the kind of team science that we're shooting for. And these are just examples of different things that we've done in society. We've done events at the Society for Neuroscience and the Society of Toxicology to try to bring these two together, and other things like this. We've had many, many workshops and so forth. All right, so in summary, most diseases can't be explained by genes alone. There should be a more holistic approach, which includes endogenous, exogenous, and behavioral factors. Many of these diseases and disorders are now really thinking seriously about exposomic factors. Complexity includes how to integrate these exposomic factors and how to interpret these factors across the lifespan. And again, we're not talking about one, maybe two, but even more than that. But we're starting with something where we're looking at just a few of the factors. So most health inequities are probably driven by exposomic factors. So I think this has lots of relevance in the health inequities part. All right, so with that, I will turn this back over to Dr. Spriggs. Thank you.
Thanks, Dave. At this time, we will transition to Dr. Erica Glasper, Associate Professor in the Department of Neuroscience at The Ohio State University. We are honored to have Dr. Glasper join us today, not only to share her exceptional research, but also because she was the first investigator that we funded in the ONETOX office. Welcome, Erica.
Thank you so much, Dr. Spriggs. So I just wanted to first thank you all for inviting me here to share with you what I've titled Researching Aspects of the Neural Exposome One Stage at a Time. So this is less a research data, empirical data discussion, and more of a how did I get here? How did I become so fortunate to be the first researcher funded by the ONETOX program. So thank you again, Dr. Spriggs, for that introduction. All right, so for those of you that I don't know, as Dr. Spriggs said, I am an associate professor in the Department of Neuroscience and resident faculty in the Institute for Behavioral Medicine Research at The Ohio State University. I transitioned here from the University of Maryland, where I started my independent laboratory some 11 years ago now. Since Dr. Jett was putting in plugs, I also want to plug our NIGMS funded post-baccalaureate research program, Discovery Prep. I'm one of the co-directors, so I clearly have a tight knit with NIH right now, and I'm happy to be here representing that program as well. I also wear another hat, proudly wear another hat. I'm a faculty mentor in 2 graduate programs here at Ohio State, the neuroscience graduate program and the biological sciences graduate program. Now, what are my research interests? What does what I do have anything to do with the neural exposome? Well, I take an approach where I study social behaviors in small rodent species, and I look at the interactions between hormones, neuropeptides, or gonadal steroid hormones, or adrenal hormones, and how they may moderate or modulate the way our central and peripheral immune systems interact with the brain. And so I was trained in psychoneuroimmunology as a graduate student, and I've taken that foundation throughout my research since that time. Excuse me. So my lab, the Social Neuroscience Behavior and Health Lab, has one large goal, and that's to understand how neuroendocrine and neuroimmune processes alter social behavior and health. And I realized as I was putting this talk together that I've been studying really the same thing for about 26 years. And all of this, my drive or my yearning to understand how social processes, social behaviors themselves can impact health outcomes started as an undergraduate at Randolph-Macon College in Ashland, Virginia. And my undergraduate research advisor was Kelly Lambert in the psychology department. After that, I quickly transitioned to graduate studies with Courtney DeVries here at Ohio State, where my training was in psychobiology and behavioral neuroscience. And in each, at the undergraduate level and at the graduate level, I was trying to understand how social behaviors, positive valence behaviors or interactions could protect the brain and the peripheral immune system to increase health outcomes in an unchallenged animal or in an animal following stroke or cardiac arrest. When I pursued my postdoctoral fellowships at Princeton University with Elizabeth Gould, focusing on behavioral neuroscience, I wanted to move from the macro understanding of brain function to understand micro functions of the brain, looking at the production and the survival of newborn cells in the brain, according to adult neurogenesis, or the enrichment of cells that we've had in our brains since birth, looking at dendritic spinogenesis. Now, I was really fortunate to get the researcher bug early in my career. So while an undergraduate, I spent 3 of my summers at Randolph-Macon, undergoing 3 different summer undergraduate research fellowships. And then as a graduate student at Ohio State, my first 1.5 to 2 years of my stint there was funded by an NINDS diversity supplement. And this really, for those of you who aren't aware, please learn more about these NINDS diversity supplements. They can pave the way for new graduate students to understand what it means to hypothesize and to generate ideas, and then to see them into their first funded fellowships like mine, which was a pre-doctoral NRSA or an F31, and it was funded from NINDS. And that lasted throughout my graduate funding. So I was really fortunate to be self-funded, if you will, for my graduate studies, and I didn't take that lightly. When I transitioned from the Ohio State University with my PhD and started my my postdoctoral fellowship, I was fortunate to not only be funded by the Merck United Negro College Fund postdoctoral fellowship, which I don't think exists anymore, which paired me with some industry scientists at our local Merck in Bluebell, Pennsylvania. But that afforded me the time to generate preliminary data to submit and ultimately get funded my postdoctoral NRSA that was funded through the National Institutes of Aging. And so as you can see, there was this nice path that was being laid that ultimately culminated in my first R01, and I'm proud to say that the ONETOX program funded my first R01. Now, for those of you who know about publishing, or excuse me, publishing papers and getting, research funded, it's not a put one application in and get one funded application. That was not my story. I am not that fortunate. But starting from the NINDS diversity supplement through my pre and postdoctoral NRSAs, they prepared me for writing the R01 and really strengthened my grant writing skills. And so I can only suggest that if you have the opportunity to pursue those mechanisms yourself. So that is how I got here. What I realized in preparing this discussion and in my talks with Dr. Jett over the last couple of years is that I've been studying the neural exposome my entire career, but most of my questions really focus on these behavioral factors, how psychosocial effects like bonding or loss or grief can really modulate our brain's response to additional stressors, and how different lifestyles, mating, pair bonding, the act of being in an enriched environment can really protect our brain from later life insults or consequences. And so as a graduate student, I know I did not know what the neural exposome was, but now I'm very fortunate to say I built my career studying one of the factors that contributes to brain function. My lab, to transition into what my lab does, we study the influences of social interactions throughout life, starting from those very early stages that have those great sensitive periods of plasticity that are open well into adolescence and start to slow into adulthood and into later life. But we know that different social factors can shape the ways in which our brains respond to later life insults. And so my research has used both hormones from adrenal steroids or gonadal steroid hormones, and even neuropeptides like oxytocin to understand how they might alter different types of structural plasticity, from spinogenesis to neurogenesis, or to the modulation of neurotransmitters. And we do so throughout life, starting early in development, as soon as one day of life, and well into middle age. And our lab is very fortunate to try to understand how these different social interactions can shape different types of plasticity via its effects on steroid and peptide hormones and immune functioning. And so right now, our ONETOX funded R01 takes a basic science approach to studying social bonding and loneliness. And I may be introducing this cute mouse species to many of you on this call, but this is the California mouse, the Paramyscus californicus. And we use this mouse to study social bonding and loneliness for 3 reasons. One, they're genetically monogamous. And so we're able to, both in the wild and in the lab, see the effects of long-lasting mate bonds. Now, that's not something that you typically see in humans. And so we can have control over the duration and the strength of these bonds. Not only do these mice form strong bonds with their mate, but they also form excellent enduring bonds with their offspring. And so because of that, we're able to look at the negative behavioral and physiological response to pair bond dissolution or breaking of those bonds. And so in my lab, along with my colleague Gretchen Nye at Virginia Commonwealth University School of Medicine, we think that by manipulating these social bonds in the laboratory, we can facilitate the examination of the impact of bonding and bond dissolution on both behavior and physiology. And so in our funded R01 titled From Social Disruption to Neural Compromise, Establishing Markers and Mediators, we take a three-pronged approach to try to understand how the neural glial response that is engaged or strengthened by social bond, not strengthened, but diminished by social bond disruption can increase neuroinflammation and compromise neural mitochondrial function. And we believe that it's through disruptions in oxytocin signaling. And our aim is to be able to predict how behavioral change or behavior before dissolution can predict how the brain, both mitochondrial function and inflammation, can indicate to us the extent to which that bond dissolution will have negative effects on neural and glial response, and ultimately being able to restore these responses via oxytocin. And so I was asked if I could give one word of advice to any of you, and you've probably heard it already. I have two words, so sorry, Dr. Spriggs, I'm taking some pleasure here. The first is to make contact with NIH early and often. I don't take for granted that my first NIH grant was a supplement to my graduate advisor's current R01. That really jump-started my my career, and I'm very thankful for the interactions and the feedback that we all received throughout that process. The second is to hold on to those networks that you build as early as your graduate training. Many of my collaborators, many of the scientists that I engage in multi-PI projects right now were folks I met as a graduate trainee. And while we may not have had the opportunity to collaborate the way we wanted to when we were still in training, once we all became PIs, we knew the strengths and the determination and the expertise that each of us had. And then we were able to bring together these partnerships that make for dynamic team science, that make opportunities like ONETOX available for us to go after because of the multi-pronged approaches. And so I just want to thank ONETOX for funding, the Ohio State, not only the College of Medicine for this, and the Center for Clinical and Translational Science for providing funding for additional work, my colleagues in the IBMR and in the Department of Neuroscience, and those that have made all of this work possible. And I will stop here.
Thank you, Dr. Glasper. At this time, I would like to invite Dr. Neil Drew to moderate the Q&A session. Please, everyone, put your questions in the Q&A box so we can get started.
Okay, thanks. Dave, I don't know if you were gonna take away the slides now. Okay. Everyone, please feel free to keep, we have some questions. Feel free to keep asking them. I'll start with a couple that were submitted in advance, and we'll go through these from there. So the first question, and this is to Dr. Jett, are there potential training opportunities in the area of the neural exposome? And what about human development research?
Yeah, I mean, I think that is something that people are starting to do. There are several programs around right now. There's something called the boot camp at Columbia, the Exposome boot camp at Columbia, which I believe is open to anyone who applies, and that just sort of tells you. And then there's other places that have sort of mini departments of I would say probably more like 3 or 4 places at institutes where they have like a little mini department of exposomic research. Some of it may be only focused on cancer, some of it may be focused on something else, but the answer to that is yes, and I think it's growing, and I think it's going to grow bigger and bigger. So I would just go and go into your someplace in one of the, what you call it, but go and try to find out by Googling exposome research, and I bet you would find a few things that are interesting.
Okay, thanks. The next question is about the grants that ONETOX is going to support. Are they going to be about understanding the mechanisms of environmental exposures on neurological disorders?
And I would say the answer, I don't want to ask any other questions, but the answer is absolutely yes. But if you were going to ask me if it was about cohorts and studying things that are not necessarily, just to see associations between this, if you want to say, I would say yes to that as well. So the answer is that we're doing both. But the thing that I always stress is that you really need, I mean, there's lots of different things that you can think of as exposomic factors, but to really hone in to ones that actually make, that actually have exposomic kinds of effects, then you need to do mechanistic research to give it more biological plausibility and towards even, causality. So I would say the answer to that is yes, we definitely, and especially with the, I think microbiome, we had a lot of great studies like that, and a few others, we had some good studies like that. They were really just looking at the, and some of it was in mice and rats and things like that, and just that, but there was already enough association data out there already, even cohort studies in humans, that it was time to really dig down and look at some of the mechanistic research.
Okay, yeah, thanks. That actually touches on the next question. And Dr. Glasper, this is for you. So when you described your research, you showed that it's, you're doing these studies in animal models. And the question is, would you expect that the mechanisms that you find are going to be conserved? I guess, first of all, as how maybe you can expand on how you see that they might translate to humans, and then in humans, would they be conserved across different groups, or how might these be influenced by perhaps other factors, other exposomic factors?
Absolutely. So the first question, oxytocin is a highly conserved neural peptide. I mean, you see it across taxa in mammals, in amphibian species, in non-human primates, and then human primates. And so we would expect, if we saw that oxytocin was driving some of the protective factors of social bonding, or preventing the stress-induced effects on microglial proliferation or changes in the activation of microglia as a result of pair bond dissolution, then we would expect to see that in humans. In fact, there is, we have data to show that, oxytocin receptors on immune cells in humans are modulated in individuals that are paired or unpaired, partnered or unpartnered, married or widowed. So it does seem like something like that is highly conserved. Now, talking about different, your second question was about different groups of people or different types of people. So not everyone will respond to the same social situation exactly the same. For example, one might think that being in a long-term partnership could be pleasant and advantageous. And then others would say, well, that seems like not a great experience to be partnered. So whether or not the mechanisms would be the same in those individuals would have to be empirically tested. And what's nice about some of these behavioral factors, if I can speak freely, is that you are able to, in humans and in preclinical research, try to tease out the weight of these behavioral factors on the outcomes. And so you can see whether or not the valence of a situation would be the same in one group of individuals or the other.
Okay, that's great. Thank you. We'll go to, so now we have some momentum of questions in the Q&A box, so we can go through some of those. So the first one, and this one, Dr. Jett, is for you. We've heard this a lot. So there are potentially many different kinds of exposomic factors and many mechanisms. So, how does a full exposomic approach fit into a single grant?
No, I mean, this is the question we hear a lot. And I always go back to the idea that this kind of came up when they started to think about the genetic genome project. It was, oh, this is a lot of genes, and I always, and this may be a cop-out to the answer, but I always say you got to start somewhere, first of all. But then the other thing I say is there's so many things that are out there right now that make it possible to try to do this, that it may be time to go ahead and start. And we may have to start slowly, 1 or 2 exposomic factors, 2 or 3 exposomic factors, and see how they interact and see if which ones are, plausible from a, as we were talking about, just plausible from those kinds of studies, and then just go for there. But I absolutely agree this is very tough. But things like, well, I don't really like to say AI, but there's lots of things that are going on now. I mean, if you think about something like wearables, if you want to look at environmental, I mean, the wearables now are just unbelievable. There's like a wearable for every piece on your body where you can, I mean, there's things you can throw up in the sky and then they float down and it gives you air pollution data, and on and on and on. So I think that it's going to be slow, but I think we're ready to go. Oh, that actually rhymes. But yeah, I understand your question, and I don't have the better answer than that. Is we've just got to start somewhere.
Okay, thanks. Well, that'll lead to kind of the next question, and that is, so because we're talking about maybe different factors maybe different mechanisms. And, so it's naturally going to require to build diverse teams. And Dr. Glasper, definitely jump in on this, because you were talking about this in your during your presentation. And is that so to build these collaborative, dynamic teams, how do you suggest that people go about finding potential collaborators, where from fields that they might not be, they're adjacent. They might not be completely familiar with everyone in there. So how do you build those collaborations to do exposomic research?
Please, I would, Chardell, Claudia, Neil, I don't want to do all of the hard work. What are your ideas? Nobody wants to try? Well, I mean, we did a RFI, it's RFI, right? Where we were looking for those kinds of answers. And we got the usual things is, you've got to first make it worth their time in terms of getting grants, where we actually ask for team science. And the other beauty is for this is that forget about the exposome, but team science is huge at almost all of these institutes. So people are thinking about this team science, and that's another reason why I think it's a good time to do the exposome. And that is saying, hey, look, we need this, we need that, and we need this. And in a lot of, I think in a lot of the ones that we've put out so far, we've made it mandatory that you have this MPI and multiple, whatever that stands for, but having these different people on board. So that's one way of forcing it. But the other way is just, as Erica was saying, talk to your friends that and say, hey, I've got a good idea. Would you like to join? And you'll be surprised now, especially how many people know that that could be a really good idea. Anybody else?
I'll just add, you have to take a risk of putting your ideas out there. I know we're in the age of, you don't want to be scooped. You don't want anyone to steal your idea. But we also realize that no one lab or one team of individuals can do the work by themselves. And so it's almost, I see it as a detriment to not try and extend the borders of my lab by collaborating with someone who may be approaching using different tools or approaches, but they're complementary. And you'd just be amazed at how many, like that'll work, or we should try the opportunities that will, it won't seem like work. For me, it seems like fun. To be proposing the stretch or the wild science. And then, the joy that you get when someone is really interested and wants to fund it so that you can pursue those questions.
Okay, that's great. And I see Dr. Spriggs put a link in the chat that's about Reporter. So that's a web tool that you can use to find funded research. Okay, so I'll bundle the next couple questions together. They're sort of related. Dr. Jed, this is for you. So other than NINDS, can you speak about some of the interest in exposomic research at other institutes, maybe at NIEHS or NIMH? Are there potentials for collaboration that way? And you can expand on it. I'll leave it to you.
No, I mean, I think you said everything. But one thing before, I think I don't want to go too fast. That Reporter is a very, very important thing you should use to look for people that are doing other things that you would like to do with them in an exposomic. So please look at that. And it has every, thing that we fund, and a lot of you know about this already, but take a look at it for those who don't, for that other discussion we had. Yeah, there's a lot of institutes, and they grow by the day now that are doing this. I will say that this has started out at NIEHS years ago, this concept. And they are, you know, a really, I think, up to speed in all of this, in this area of the exposome research more than anybody, but they can't do it all of themselves. And, you know, they may not have, I mean, if you think about it, think about the neural exposome. Yeah, there's some great 1 or 2 people there that work there at NIEHS and know some people in the, but think about the thousands of experts that we have at NINDS that aren't doing this research because they don't know a lot about it. And so this is why it's important for this to be a, this should involve all of the ICs that have that experience in some of these exposome factors. So I'm very happy to say that people like NIA for Alzheimer's and Alzheimer's disease related dementias and other things, doing a great job on exposomic factors. National Institute of, I would say probably NCI is first after that, cancer research, and on and on really, for the NIDDK, for the microbiome. So all of these, I think, maybe not all of them, but many of these other ICs are catching on and thinking about this concept of, so there's a lot, exposome. So there's a lot going on here at NIH. And I'm glad we're one of them, ICs.
Okay, that's great. So the next question, I'll jump back a little bit to these questions of collaboration. And so this I'll put to Dr. Glasper. If there's a student or a potential trainee who is interested, how would you recommend that they approach faculty? Maybe they're interested in their research, potentially to join the lab, although is that necessary? I guess what would be your recommendations?
So if you're a current training, like a rotating student or trying to get into graduate training, I think, so some programs, let's just be real, some programs say, oh, it's not necessary for you to reach out to the potential PI. They won't have time to read your emails. But, as a PI, as most PIs who have very thin skin, you kind of like it when somebody reaches out to express the interest in your work and to ask you if you are accepting new students. What if you're in a program and you have an idea that would be a good way for your current PI and another PI to collaborate? Most times, we as faculty are looking for ways to bridge relationships with our colleagues, and you kind of need warm bodies, willing people, interested individuals to do the work. So you put the feeler out there. You'd be surprised how many collaborations here at Ohio State start. It's usually because within my institute, the trainees are buzzing around and cross-pollinating. And so then it gets to us and we're like, oh, that would be really cool. Oh, we should, put in a small seed grant for that. Oh, that falls under this purview. Oh, that's a nice pilot project. Put yourself out there, because if you're thinking about going down this line, of like going from trainee to independent scientist in any realm, you're going to have to pitch your ideas. And sometimes you'll have 3 minutes to do so, and other times you'll have 12 pages, but you still have to pitch your ideas. And if you hold on to them, then nobody's going to know that you're really interested. And so I just would not hesitate to do that. And I know it's easier said than done. And I am someone who is very introverted, and I have to overcome that first hesitation to not make that contact and introduce myself, because it's those things that have led to these team science projects that I've been afforded opportunities to participate in.
Okay, thank you. Yeah. Okay, Dr. Jett, the next question is for you. I'll join these together. These are again about the kinds of research that NINDS and ONETOX would like to fund. So you showed us some of the funding opportunities that have been open in the past or that are currently open. So it seems a lot of these are on Alzheimer's disease and Alzheimer's disease related dementias. Is there a plan for ONETOX to fund research into other disease areas, maybe pain? Another one could be about, it's just, I mean, I'm just reading from the question. The role of maternal or early childhood exposome, how that has a role in neural development. So, and if that does become part of the plan, so what should people do? Just look for keep scouring the pages in order to find, an RFA or something that comes out?
Well, that's a great question. And I was smiling because yes, we do have a lot going on with AD-ADRD, Alzheimer's disease and related dementias. Yes, but the answer is absolutely. We're covering everything within our mission space. And what's in our mission space is, I mean, a lot. We're not covering all 800 diseases, but a lot of eventually all of the major things that, we're working on, like Parkinson's and stroke and all sorts of things that, I mean, you can find on our website, and you'll see from the pages some of the things that we're really thinking about right now. But even then, if you've got a good, if you've got a good premise for a grant for things that you don't even see, but that they're within our mission space, that's good to go as well. And with that, hopefully you'll follow what I'm saying. So with that, most of the things you saw that I showed were specific asks from you. We need you to do this. But that does not mean you can come up with your own idea and just submit this as an R01. And that will just, and I'm really, really, really want to see people doing that. Okay? Forget about the things that you see are fun, we're looking for funding. Just come to your own thoughts and just talk about an R01 or R21, and then, or one of these, but it doesn't have. Now, with that said, we are now in the process of developing a strategic national plan. So we're holding them back a little bit from answering those kinds of questions until we have the final plan that is hopefully going to be soon, where we have the experts of experts tell us what we really need to do. But with that said, I know that's two things, I'm saying two things. With that said, still, if you have an idea, let's talk about it. And don't be afraid to submit a proposal.
Okay.
Not too much? Not enough?
No, I thought it was great. Okay, so we have a couple questions left. I know we're also getting to the top of the hour. So I will just urge you that we'll go through these last couple questions and then we'll be done. So if you have any, if you've been waiting for someone else to ask your question, it's not going to happen. You have to ask your question now. And so, you might want to type it. Okay, so the the next question is, this is for Dr. Jett. Can you describe some of the clinical work that is either funded or could be of interest to ONETOX?
Yeah, we've got a great clinical body here at NINDS. I think one of the best. So the answer for that is yes, we do that. A lot of it that we've been doing in my sort of space are things like this is human studies, association kinds of studies. What's the association between radon and stroke? Okay, or how does isolation affect people after stroke? Or things like, you know, why is bonding? How does bonding affect brain function? Our best ever one, I'll say that. So and that's not really a clinical, but in a sense it could be. So a lot of what the answer is we still have a lot of work to do on which of these exposomic factors are relevant to health in people before we start digging into some of the other kind of research. And we're getting there. I'm very happy that the leader of, I'm very happy that the folks here that do clinical research are on board, they get it. And we're going to hopefully do more of that research. And again, if that question is about, should I even bother talking to NINDS because I do clinical research? The answer is absolutely yes. And I will get other clinicians on the phone.
Okay, thanks.
You know who asked me that question? One of my sons, who's an MD. He says, wait a minute, Dad, are you going to be going? He said, yes. But go ahead, sorry.
Okay, well, the next question, Dr. Jett, is for you. So DOD started a program on environmental factors and Alzheimer's and psychiatric disorders. Is there a potential for collaboration on that? I guess collaboration on DOD funding opportunities?
Well, yeah, we like to have things like that. But right now, I don't know of any. Well, I actually did have, I was on the committee for one of those kinds of studies recently. But yeah, we're always open to collaboration, and especially with DOD and, you know, CDC and all the other ones. So we're hoping to do more of that. Right now, I'm not seeing anything in the horizon, but if you know about anything, let's talk.
Okay. All right. So the next question, I think, can be for both Dr. Jett and Dr. Glasper, and that is, what role do you see technology and technological advances playing in this area of research?
I'll let Dr. Jett take that, because you spoke moments ago about wearables, and so maybe that is a good segue.
Fantastic. I mean, oh, wait a minute. Do I? Yeah, I think I have this figure. Oh, well, we're not sharing screens anymore. So I have this famous real figure where it's a person standing and every part of their body has wearables, you know, from your finger to the, so I think that that's huge. And just big data, you know, machine learning, I don't even like saying the A word anymore, AI, but machine learning and other things that are able to, and there's one able to handle all of the data. And one thing I was going to say is that at NIEHS, they've got a great program to try to look at some of these exosome factors and put them together in a way. So that's something that you can look at. It's an advertisement. It's not anything new, but you can go to their website and see what they're doing. And other ICs are doing that as well. Yeah, I mean, I think that there's enough, at least, maybe we're not there yet, but there's enough to start doing these kinds of complicated, mega data kinds of studies.
Okay, yeah, I'll also just mention, I think if someone is interested in machine learning, and maybe they're not interested in being in neuroscience or science long term, but this can be a very interesting introduction to data science, and then you can go on and sell advertising clicks or something like that later, but can learn the tools of the trade right here. Okay, there's I think 2 last questions. One that I'll ask, it's come in, is it says, for someone new to this area of research, it can be hard to generate preliminary results. So are these, how important are these for, let's say, an R01, or, are there maybe some other grant mechanisms that would be more suited for, a new investigator to this area of research?
Oh, sorry. Did I turn on the, am I sharing the screen?
You're sharing.
Does it have these opportunities, exposome research?
Yes.
Yeah, here's some tools that I was thinking about. You can take a look at for that previous question. So this question was about different kinds of, what was the question?
The one that I just asked you was
Yeah.
about how important is preliminary data.
Oh, that's important in all research here at NINDS, at NIH. And I understand why that's a question, because a lot of it is very young kinds of research, is to just see if there's any relationship between whatever and whatever else. So I have to be careful about things that I'm thinking about, but that's something we recognize, and hopefully we can do something about. In the meantime, there's R21s, right? And R21s usually don't ask for as much or even any preliminary data in some cases. And I know, but anyway, that's out there. And then the other thing is some of the other things we can use for more exploratory kinds of projects. So that's something we're thinking about. We do understand why that's an issue in this kind of research.
Okay. Thank you very much. And with that brings us to the end of the questions. There's, if you have any further questions, so Dr. Spriggs put this in the chat. If you have questions that are, about your specific scientific research, we encourage you to reach out to us. If you have even at general questions, please reach out to us. So that'll end the Q&A, and I'm going to pass this back to Dr. Ebrahimi.
Thank you. Dr. Spriggs, did you want to say anything, or should I close this out? Okay, great. Well, thank you all so much for attending today. Thank you so much to Dr. Glasper for sharing kind of the peer side of things and your experience with NIH. That was truly so valuable. I hope everyone found this valuable, to see the people behind NIH, the people that will be helping you hopefully with these grants. And as a reminder, we will be sharing the slides, the recording, all the emails, so you'll know exactly who to contact, all the resources, everything, probably by the end of the week. So look out for that email very soon. I'll also post in the chat the diversity listserv that we have. So we really encourage you to stay connected with us, subscribe on the right-hand side of that website. And so you'll hear about more events. Like Dr. Spriggs mentioned, when we have the website up for the strategic planning of ONETOX, we'll share that with all of you. But thank you all again for attending, and we look forward to staying in touch. All right, take care.
We'll see you.